Good evening, everyone. Welcome to Cell Talk presentation activity. For today's Cell Talk presentation, Diotima is the presenter and she is going to present on the topic Importance of Study of DNA Protein Interaction in Disease Mechanism Research. Diotima is a research scientist at Renix Life Sciences. She is presently working on multiple projects which includes data acquisition, gene maps, and she holds an MTech in bioinformatics from Maulana Abdul Kalam Azad University of Technology and BTech in biotechnology from Techno India University. Diotima aims to utilize her expertise in bioinformatics to support research in genomics, proteomics, and drug discovery. She wants to expand her work with a focus on tasks such as analyzing high throughput genomic data, developing computational pipelines, and contributing to the development of novel therapeutic strategies. She seeks to drive impactful solutions in healthcare and biotechnology while continuing to advance her technical and scientific acumen. I welcome Diotima to give her presentation.
Hello everyone. So I will be discussing the topic importance of study of DNA protein interaction in disease mechanism research. Moving to uh, introduction, DNA protein interactions mainly refer to the binding and interaction between protein and DNA molecules. Uh, this plays an important role in various biological processes. There are mainly two types of DNA protein interaction. One is sequence specific DNA binding, the other is sequence non specific DNA binding. In sequence specific binding, the protein binds on a site having specific nucleotide sequence, and for sequence non-specific DNA binding, the DNA binding protein can bind to a DNA in a random position in the sequence of DNA. In uh, for like the DNA specific sequence specific DNA binding, the interactions are mainly maintained by hydrogen bonds, uh, ionic interactions, and vulnerable forces. Uh, the here, in case of transcription, the transcription factors are a special kind of DNA binding protein. They can recognize only a specific DNA sequence. For sequence non-specific DNA binding, uh, that is during replication, the DNA double strand is maintained by helicase enzyme and the replication fork is made. A special kind of protein called single strand binding protein binds to the melted single strand DNA and stabilizes the system by preventing them to be denatured. So, the binding of protein to specific sequences or structural features of DNA allows them to influence the activity of genetic material. For example, the transcription factors bind to the promoter regions of the DNA to initiate or repress transcription, while enzymes like DNA polymerase are crucial for replication. Additionally, histones interact with DNA to form nucleosomes, affecting DNA packaging, gene accessibility. These interactions are typically characterized by specific recognition sites on DNA, such as motifs or consensus sequences, where proteins binds with a high affinity. Understanding these interactions is crucial for studying disease mechanism. These interactions uh, play a crucial role in several molecular and cellular mechanisms. Studying these interactions helps in understanding the uh, uh, underlying mechanisms that contributes to the disorders like contributes to neurological disorders. Moving to next slide, that is techniques. So the some of the techniques to study these interactions are chipsec, DNSec, CRISPR's based tools, visual, the icons representing lab techniques. So, here chipsec is uh, described here. Uh, for chipsec, the DNA protein complexes are cross-linked here and using formaldehyde to preserve their interactions. Chromatin is then sheared into smaller fragments by sonification or enzymatic digestion. Uh, for immunoprecipitation, antibiotic specific to target protein is used to pull down the protein DNA complexes. This isolates only those DNA fragments that are bound to the protein of interest. Uh, for purification and for reversal of cross-link, the DNA protein complexes are purified and the cross-links are reversed to uncover the DNA. The library preparation and sequencing, the extended DNA is used to construct a sequencing library and is uh, used for to sequencing to uh, used in high throughput sequencing. Uh, Last is like data analysis as sequencing reads are aligned to the reference genome. The peaks representing the regions of DNA binding proteins are identified using computation tools. The mapping binding sites of the chipsets are used to map the binding sites of transcription factors and other DNA associated proteins, and also used for studying histone modifications to understand epigenetic regulation and also to investigate changes in DNA protein interactions in response to stimuli or in diseases. Moving to the next slide, role in disease mechanisms. Uh, the DNA protein interactions are important in pathology of genetic disorders like cancer and neurodegenerative disorders. Yeah, in these like genetic malfunctioning mutations in coding regions disrupt the formation of proper like functioning protein complexes, causing dominant negative effects on diseases linked to the faculty protein assembly. In cancer, dysregulation of DNA and RNA binding proteins leads to the abnormal gene expression and metabolism. Uh, in For neurodegenerative diseases like Alzheimer's and Parkinson's, the the DNA protein interactions cause misfolding and aggregations. Moving to next slide, like how protein interactions damage and causes neurodegenerative diseases. So these interactions play a pivotal role in the pathogenesis of these diseases. The proteins like uh, tau, beta amyloid, and alpha synuclein interacts with DNA, influencing gene regulation and contributing to disease mechanism. Uh, this DNA damage and chromatin rearrangement often involves tau and alpha synuclein and are often linked to the neurodegeneration, suggesting potential therapeutic targets in these areas like for DNA repair and chromatin reorganization. Uh, like for cancer and autoimmune diseases, the DNA damage responses maintain genomic integrity by repairing DNA or triggering apoptosis. When DNA damage is like inevitable, uh, the defect in DNA results in mutations that drive tumorigenesis. The methylation changes caused by DNA protein interactions play an important role in gene regulation and cancer development. And for autoimmune diseases like systematic lupus erythematosus, the DNA protein interactions are linked to immunodysfunction. The like diseases like or interactions like mannose binding lectin oligomers are associated with disease activities, while the antibodies play a crucial role in systemic lupus erythematosus pathogenesis. Looking at the therapeutic potential of DNA uh, protein interactions, the advancement of therapeutics to DNA protein interaction studies is a transformative approach in cancer therapy. Uh, for targeting protein interaction in cancer therapy, DNA damage response pathways maintain genomic integrity, but they are often dysregulated in cancer. Targeting protein interactions within DNA damage pathways using small molecule inhibitors selectively kills cancer cells or enhances the traditional pathways of therapies. It enhances the efficacies of uh, traditional therapies and for predicting drug interactions with DNA binding proteins. The network based models predict interactions between DNA binding proteins and drugs, like clustering binding sites based on physicochemical properties. For example, erythromycin interaction with transcriptional replaces highlights opportunities for targeted drug design and for high throughput interaction studies. Technologies like DNA curtains enable high throughput real time analysis of DNA protein interaction at the single molecule level. These platforms aid in localizing the interaction essential for developing nanotherapeutic or nano devices and therapeutic strategies. Coming to conclusion, uh, it can be said that importance of uh, uh, DNA binding interactions are fundamental uh, to study different interactions which can uh, pave pathway to understand the underlying mechanism for diseases. Also, there is a lot of opportunities in therapeutics and clinical studies. The future researchers may implement AI and other technologies which can bring breakthroughs in this area. These are the references that are used for this presentation. Thank you everyone for listening.
Thank you, Diotima, for your presentation. Audience, now the floor is open for any queries. If you have any questions, you can type it in the chat box. Okay, I don't see any questions. So, Diotima, can you move to the last slide? Yeah, next Cell Talk presentation will be by Anshman Roy, and he will be delivering the topic use of AI assisted image processing in healthcare. And the presentation will be on 29 Jan 2025. Thank you everyone for your attendance for this Cell Talk presentation, and I hope to see everyone in the next presentation.